Hello everyone, welcome to JASCO's webinar. My name is Taijo Yama from JASCO Corporation. I'm in charge of developing new applications for biopharmaceuticals. It's my great pleasure to share our cutting-edge application for therapeutic antibodies. I will first briefly touch upon the feature and recent trend of CD spectroscopy, and then introduce these technologies: accurate secondary structure estimation, automated CD system, statistical evaluation software. CD spectroscopy employs the circularly polarized light. The light we generally see in nature has random surface of oscillating electric field. On the other hand, the electric field of linearly polarized light aligned in a single direction. Linearly polarized light can be decomposed into two these circularly polarized light. The direction of oscillation vector of the electric field changes clockwise, called right circularly polarized light, and counterclockwise, called left circularly polarized light. Circular dichroism is a difference in absorption of these two circularly polarized lights. Compound that has chirality, such as amino acids and sugars, exhibit circular dichroism. Protein composed of amino acids and nucleic acid composed of ribose also exhibit dichroism. And this CD spectrum show a unique shape depending on the conformation of protein, peptide, and nucleic acid. So what are the advantages of CD spectroscopy? There are some structure determination methods such as X-ray crystallography, NMR, and cryosem. Although CD spectroscopy could not offer such high-resolution information, it has these unique advantages. CD spectroscopy offers robust data on biomolecules at low concentration. We simply set the cell and run spectrometer, and we can obtain one spectrum in about 5 minutes. CD spectroscopy allows the measurement under various conditions such as temperature, pH, and with various additives such as salt, ligand, and chemical denaturant. As the market of therapeutic antibody grows, the number of publications is consistently increasing, and the use of CD spectroscopy is expanding in the academic field and industrial field. CD has long been widely used for protein structure analysis, mainly in the academic field. The development of these game-changing technologies, high-throughput system, accurate secondary structure evaluation algorithm, and statistical similarity evaluation method has accelerated the use of CD spectroscopy to biopharmaceutical companies. First, I'd like to talk about on the secondary structure estimation, SSE, and its application. So what information does CD include? Protein CD spectra are generally measured in far UV and near UV region. Far UV region contains information of secondary structure, and CD spectroscopy has been widely used for analysis of secondary structure. Near UV region contains information of side chains of aromatic amino acid, like phenylalanine, tyrosine, and tryptophan. Although the signal only about 1/100th of pi UV, these side chain CD signals are sensitive to their molecular environment and tertiary structure. So the near UV CD spectrum makes it possible to evaluate the structural similarity like fingerprint identification. Near UV CD measurement tend to be used for similarity assessment for antibody drugs in quality control process. And secondary structure estimation was first conceptualized by Fassman and Greenfield in 1960s. Then, in 1990s, relatively earlier than in other scientific fields, the machine learning was introduced to improve estimation accuracy. However, challenges have been lying ahead for a long time, and now BeStSel algorithm breaks through it. Here's an example of typical 5-VCD spectrum. Alpha helix show a positive signal around 190 nanometer and negative signals at 208 and 222 nanometers. Typical beta-seed-rich protein tend to show a positive signal around 200 and negative signal around 217 nanometer. Random coil-rich protein like trypsin inhibitor intrinsically disordered proteins, IDP, show a negative signal around 200. CD spectrum show a unique shape dependent on the secondary structure of protein. And the secondary structure estimation, SSE, is a method to estimate a fraction of these secondary structure elements using the shapes of the CD spectrum. The principle of conventional SSE is simple. The protein CD spectrum is assumed to be a linear combination of these base spectrum, helix, seed, turn, others. And the fraction F are calculated so that the sum of these base spectra most closely matches the measured spectrum. This method is called CLS, and we JASCO provide as a JWSSE. The CLS method has major challenges in estimating the beta seed. The spectral intensity of beta seed is considered small compared to other base spectrum, like helix. So CLS does not work well for analysis of beta seed-rich protein like therapeutic antibodies. This bar chart shows the results of SSE for immunoglobulin G. Dark blue is the results of SSE, and light blue is from X-ray structure. It is the answer. In particular, the beta seed and others show significant differences between SSE and X-ray. So various machine learning methods were applied to overcome this situation. And a machine learning approach, model protein with the correct secondary structure fraction were used and create learning model by what CD spectra show what secondary structure fraction. PCA and PLS are well-known method, and we provide as a JWMBS. Applying the machine learning model estimation error seen with the CLS improved and show good estimation accuracy even for the immunoglobulin G. However, there needs to be a solution. The spectra recalculated from the estimated fraction and base spectra do not match the experimental spectra. If both the estimated fraction and base spectra are correct, linear combination of these data should reproduce the measured spectrum. However, significant difference is observed for beta-citrate protein. What is the reason for this inconsistency? The answer is the diversity of the spectrum of beta-cit compared to the helix-rich protein. The beta-cit-rich protein shows diversity of city spectrum, and all conventional SSE define the spectrum of beta-cit as a single shape, so this assumption was not appropriate. Professor Joseph Cardosh at Roland et Bosch University found that the diversity of CD spectra of beta-cit is caused by the twisting between the beta strand. The beta-cit show a seed-like folded structure of peptide chain. However, from a different view angle, the beta strands are actually twisted between the strand, and Joseph Cardosh further classified beta strand into 3 types according to the degree of twisting angle and defined their new CD spectra. So BeStSel has overcome the challenges to perform SSE based on this new definition. Specifically, by taking account for twisting, BeStSel provides 8 secondary structure elements, which is double than conventional SSE. BeStSel provides a detailed and accurate secondary structure fraction, not only beta-cit-rich protein, but also a wide range of protein. In addition, experimental spectra and recalculated spectra show great agreement. Of course, the sum of fraction of divided beta strand means fraction of normal, conventional beta strand. So, and you can compare the previous reported value. In particular, these samples for which machine learning could not offer good results were significantly improved by using BeStSel. And BeStSel is now cited in over 1,000 papers, and a lot of academic researchers use BeStSel via free web server. However, researchers at biopharmaceutical manufacturers could not benefit from BeStSel due to security issues. They cannot upload their data for website, Now, in response to their voice, Roland at Rush University and JASCO have collaborated, and we could finally develop BeStSel integrated into Spectra Manager. User can use BeStSel not only in offline environment, also user-friendly and seamless analysis platform. Of course, we could offer CFR and GXP compliant software. From here, I'd be happy to share the application that the detailed tracking of conformational changes of antibody drug using BeStSel. We selected Herceptin, the innovator for the breast cancer. And as you well know, the active pharmaceutical ingredient of Herceptin is human IgG1. And we compared Herceptin and IgG, that is the region for research. And these are the thermal denaturation spectrum of Herceptin and human IgG from 30 degrees Celsius blue to 85 degrees Celsius green. At 30 degrees Celsius, the spectra of both samples was similar, but as heated, they changed to different shapes. And to analyze these structural changes, SSE was performed using BeStSel. And this horizontal axis is temperature, and vertical axis shows estimated fraction of seed and others. This fraction at room temperature was consistent between the Herceptin and human IgG and the crystal structures. The fraction of beta strand decreased, while the fraction of others increased with heating for both Herceptin and human IgG. Tm for Herceptin and human IgG is estimated from these curves were consistent with previous reported value. The advantages of BeStSel can offer fraction of each secondary structured element, including 3 types of twisting of seed. And these are significant differences between the Herceptin and IgG in relaxed beta strand. Herceptin shows a decrease of fraction as temperature, while the fraction of IgG was maintained. So, BeStSel is useful in understanding the detailed mechanism of structure formation in protein, including antibody drugs. Then I will present an application using automated CD spectrometer and SSE. We have developed HTCD Plus, fully automated sample loading, spectrum scanning, and flow line flushing. HTCD Plus can accommodate 2 microplate, 192 samples with a volume less than 100 microliter. In addition to CD measurement, HTCD Plus can offer accurate absorbance spectra and intrinsic fluorescence spectra of protein automatically. Here we evaluate the pH stability of antibody drugs by combining the HTCD Plus and BeStSel. In addition to the Herceptin, we selected the rheanine at the sample. The left side is the result of Herceptin, and right side is the RNA. Upper figures are CD, and lower figure is the intrinsic fluorescence spectrum. Spectra of pH 2 to pH 10 are overlaid, and this kind of measurement takes a lot of time and effort by manually. But all we have to do is to set the sample on HTCD Plus and just leave it overnight. From the CD spectrum, we evaluate the secondary structure changes by pH. The vertical axis shows a delta fraction. That means the difference from the fraction of pH 7. No significant differences were observed for each antibodies in the pH range from 3 to 10. But pH 2, the three-component helix strand and the other show significant differences. This result is reasonable because typically antibody drugs are exposed to pH 3 environment through the manufacturing process in order to remove contaminated virus. Here is the result of peak shift analysis from fluorescence spectrum. Each peak position was calculated by the barycentric mean for robust variation. Both Herceptin and RNA show the same trend with a red shift of peak as pH decrease, suggesting that the tryptophan is changing to the hydrophilic environment. At pH 2, tryptophan of Herceptin was found to be more hydrophilic environment. Maybe structure was denatured, whereas very interestingly, RNA was found to return to a hydrophobic environment. The secondary structure fraction show a same trend among these antibodies, while the tertiary structure show the opposite trend. I like to emphasize that the accurate secondary structure evaluation method and high-throughput system enable us to conduct a comprehensive evaluation of secondary structure and of therapeutic antibodies. So let's move on to the next topic. I will introduce qHOS software that enable the statistical similarity evaluation of spectrum. For a long time, similarity, equivalence, or significant differences between the spectra have been conducted by visual inspection. The International Council for Harmonization, ICH, Q6B guideline recommend CD spectroscopy as one of the methods for physiochemical characterization of biopharmaceuticals. In addition, there is a requirement for an objective assessment of structural similarity described in ICH Q5E. The FDA's guidance touch upon the objective and statistical evaluation of analytical similarity between the biosimilar and their innovators. So there is a growing demand for a process to objectively determine the similarity equivalence of CD spectroscopy. For invariant data, such as this bar chart, the objective and statistical evaluation are easy. Researchers have widely introduced the judgment based on the t-test. If we like to apply this statistical testing for spectrum, it is not easy because spectrum is composed of multiple data. So in response to the demand from the biopharmaceutical industry, we developed the qHOS. qHOS can statistically determine the significant differences between two spectrums, considering the various error factors, including spectrum noise and sample preparation error, and so on. For the statistical testing, most popular student t-test and Welch's t-test are implemented for judgment of significant differences. In addition, qHOS implements tests that allow to claim equivalence. In addition to CD, qHOS can line the FTIR, fluorescence, UV-Vis, and Raman spectrum. Of course, CFR compliant software is also available. In this section, I will explain how to determine the similarity or significant differences of two spectra in qHOS. In the spectral comparison procedure, it is necessary to convert the spectral differences into one parameter. qHOS applies the Euclidean distance method. While there are various methods for quantifying the spectrum differences, the reason why qHOS applies the Euclidean distances is its sensitivity. Euclidean distance is the most sensitive method. And in Euclidean distance, larger distance means the larger spectrum differences. Conversely, when the spectra are perfectly matched, the distance shows 0. Thus, the spectra can be converted to a single value, but it is not clear how much differences mean the significant differences. Therefore, qHOS incorporates the procedure to reflect the variation of the reference and samples. The procedure split into 3 parts: input spectrum, creation of the distribution of distances, and t-test. The qHOS requires multiple reference data to consider measurement error of reference. In case of student t-test, one sample data is required. No need to measure sample repeatedly, but measurement error of sample is not considered. In case of Welch's t-test and test, multiple sample and multiple reference are required. This method can consider both reference and sample error. In this section, I explain in case of Welch's t-test. First, calculate the mean spectrum of reference, which serves as 0. Then calculate the Euclidean distances between each reference and the mean spectrum. In this way, the distribution of reference distances reflect error factors such as noise. Then calculating the weighted Euclidean distances between the sample and the mean spectrum as well. Assuming normal distribution for this, the t-value is calculated mean and standard deviation of references and mean and standard deviation of samples. Then p-value is calculated from t-value and t-distribution. And you can think the p-value as a probability of the two samples is the same in case of Ray's t-test. So low p-value means the difference. Finally, the significance is determined if p-value lower than significant label alpha. Generally, alpha is set to 5%. And here, let's think about the p-value greater than 5%. So there are no significant differences between sample and references. In this case, can we say that two samples, reference and samples, are same? That the no significant differences equal to same? The answer is no. Ray's t-test can only detect the differences. So when we claim the same or equivalence between the two samples, another testing method should be applied. That is the toast. So toast is well suited for the development of biosimilars, because toast allows for the evaluation of the equivalence of biosimilar to its original product. So let me introduce one of the applications applying toast to compare the equivalence between innovator and biosimilar. In this slide, we use not only CD, also consider the employment of FTIR and Raman spectroscopy. So regulatory authorities recommend the orthogonal assessment of biosimilar quality attribute using the multiple instruments based on different principles. 5UV-CD and FTIR spectroscopy are orthogonal method for evaluating the secondary structure, and near UV-CD, Raman, and fluorescence spectroscopy are orthogonal for tertiary structures. So a comprehensive orthogonal assessment using this method can provide a firm structural foundation to claim biosimilarity. So we JASCO have HTCD Plus for far UV and near UV CD, and FTIR for X for infrared measurement, and NRS 4500 for Raman spectroscopy. And the data obtained from this instrument were analyzed on a single platform, qHOS. In this case, we focused on 3 antibody drugs. BeStSel and Riani have same amino acid sequence and formulation condition. Both antibody target CD20 and Herceptin, which target HER2, have different primary structure in 5UV region for rituximab and different formulation condition. First, we tested are there significant differences between the tertiary structure of BeStSel and Herceptin. Although difference between the antibody drugs is not large, the shapes of near UV-CD spectra differ dramatically. Similarly, the distribution of distances shows significant differences, and p-value from the toast show 1.0. So there are no equivalence. Next, we tested the similarity equivalence of secondary structure of Riani and BeStSel. The left side showed a 5UV-CD and the right side showed FTIR results. The spectrum of Riani is in excellent agreement with that of the innovator of BeStSel, based on the 5VCD and FTIR results. And the distribution of distances are close each other, and the p-value is lower than significance label of 0.05. They are equivalence. This slide shows the results of tertiary structures, near UV-CD spectrum and Raman spectrum. Raman spectra contain information of phenylalanine, tyrosine, and tryptophan environment. Similar to secondary structure, tertiary structure of MAPTERA and RIANI show excellent agreement in both near UV-CD and Raman spectrum. And distribution of distances is close to each other, and the p-value is lower than 0.05. So there are equivalence between the BeStSel and RIANI. So we can evaluate the structure equivalence in the secondary structure and tertiary structure. So these three techniques have advanced analysis of higher order structure of therapeutic antibodies. So we JASCO continue to provide a solution for higher order structure in the future. Thank you for listening.